Dear colleagues, welcome to our monthly dialogue. Now you know what dialogues are proposed. They are very entertaining. We invite top cardiologists from the 5 continents, and we discuss with them their scientific achievements and their personal history, which is typically very inspiring for younger colleagues in particular. You know that our dialogues are given every month, is typically on the last Thursday of each month at 17 Central European Time. And they are then live streamed in YouTube and Twitter and posted in YouTube and Twitter. So if you miss the dialogue anyway, you can see the dialogue in the future. Today is a very, very interesting dialogue. We have a great guest. The title of this dialogue is From ISIS to UK Biobank, the history of cardiovascular trials and epidemiology with Rory Collins. I'm hosting Rory. Sorry for the telephone. I'm calling from China. I'm in the hotel and I'm a bit under pressure, perhaps. I apologize, but quickly I ask Perry to introduce our famous guest to start the dialogue, and which will then continue with questions and answers, and hopefully many questions from all of you. Please, Perry.
So thank you, Filippo. So it's a real pleasure to welcome Professor Rory Collins to today's EHJ Dialogue. Rory is a medically qualified epidemiologist who has dedicated his career to the prevention and treatment of cardiovascular disease in large international population-based studies. He's also the founding head of Oxford University's Nuffield Department of Population Health. Over the past 40 years, he's conducted many pivotal randomized trials, which have shown unequivocal benefits from thrombolysis, as well as preventative strategies designed to reduce LDL cholesterol with statin therapy. And as I'm sure we're all familiar, we use the results of the trials that he steered in our everyday practice. In 2005, the Wellcome Trust and the Medical Research Council in the UK appointed him as the principal investigator and chief executive of the UK Biobank, a project that involves more than 500,000 participants, and which is the largest deeply characterized prospective global study for any type of health research. More than 30,000 researchers worldwide now use the UK Biobank, generating more than 3,000 papers in 2023 alone. So Rory, it's a real pleasure to welcome you here today.
Well, thanks very much for inviting me. It should be fun. Looking forward to it.
Right, so Rory, so I did a bit of background on you before this event, and I was interested to see that when you were at school, maths was your thing. So how did that influence your career choices?
Well, I went to the careers officer at the school I was at in South London and asked what kind of career could I have with a maths degree. And they said, well, you could become an actuary. And when I looked at what an actuary did, I thought that sounded a pretty tedious career. So I was chatting to a friend and he said, well, I'm thinking about going to medical school. And so I thought, well, I guess I could do that. But then in the UK, you do your A-levels, your final year exams, and I went off hitchhiking around Europe and came back and opened up the envelope with my results, and they were far better than I thought they would be. And so I thought maybe I could try for Cambridge and do natural sciences and do maths in the first year and then see how that went. But I failed to get in. So I went back to plan B or plan A and went to medical school to St. Thomas's Hospital. And again, in the UK, you do 2 years of preclinical, and then you have an option of a third year to get a bachelor's degree, or you can go straight into clinical. And again, friends were going to be doing a third year in physiology or biochemistry or anatomy. And I thought, well, maybe this is my chance to do mathematics. And so I went around the different colleges in London, and they said, no, you couldn't do that. You could do anatomy, you could do philosophy, psychology, but not maths. So I went to the library, and for some reason, I'd got the idea that America was more flexible than Britain. And I looked up 36 universities in the US, some I'd heard of, some I hadn't, and wrote a letter to them saying, could I transfer from London University to your university, do a full year of statistics and get a degree and come back? And I think Harvard sent me a letter saying, send us a check and we'll think about it. Many didn't reply, but George Washington University in Washington, DC said, yeah, why not? And so I arranged to go, and I nearly got kicked out of medical school at that point because they said, what has mathematics got to do with medicine? But I was very fortunate. The head of community medicine there, Walter Holland, was much more supportive, and he helped me to get the general support you get from the Medical Research Council to support that. But more importantly, he found me a job to work 1 day a week in the Biostatistics Center that had been set up by Jerry Cornfield, who had set up the cancer statistics in the NCI originally and who had now retired, working in his clinical trials unit 1 day a week, and that would pay my fees. And that was my introduction both to statistics in 1976, 77 in Washington, DC, and also to randomized controlled trials, because I was being paid to work on a trial there. And that's how I kind of learned about the whole idea of randomization.
Mm. You came back. You came back to the UK. What happened then? Well,
I really enjoyed it. I really enjoyed the statistics. And so I applied to do the master's at the London School of Hygiene and Tropical Medicine. But I didn't tell the medical school. So I was trying to do my clinical at the same time as doing a master's in London School of Hygiene and Tropical Medicine, but people found out. So I was allowed to keep on going to the lectures at London School, but not actually take the degree while I finished my clinical training. Did that, and then I was doing a house job, my 6 months of house surgery in St. Thomas's, and I thought maybe this was the time when, once I'd finished that I could do research for a year or so. And I'd read this fantastic book by Peter Armitage, who had been at London School of Hygiene and Tropical Medicine about clinical trials. And I thought, well, nothing ventured, nothing gained. I found his phone number and rang him up. And he'd moved to Oxford. And I went down to meet with him. He was an extraordinary, lovely man. And he rang up Richard Peto after he'd heard what I was interested in doing, which was actually being involved in clinical trials. And that was how I was introduced to Richard Peto. And that led to me, after I'd finished my house jobs, coming back to Oxford to work on trials.
I think we're starting to learn about your career. It's a matter of sort of having a bit of chutzpah, isn't it? Sort of following the path that you set yourself. But it's also about mentorship, isn't it? And perhaps you had the timing was really good for going into this field in Oxford at that time. You had Richard Peto, Salim Yusuf was there, Peter Slight was there. I mean, how important was that mentorship to what subsequently happened in your career?
I don't know whether mentorship is the right word. It was sponsorship. It was being lucky enough to find people that believe in you, much more than you do in yourself, and who support you. I went to Oxford in 1981. I don't think I had a public until 1987, 88. So throughout that period, being supported went way beyond mentorship. And I was very, very lucky. I mean, Richard Peto had this idea for a long time. And you can go back to the 70s and find these obscure papers by him about the need to do much larger randomized trials in a whole series of areas. And Peter Slight and Salim had started talking to him about the idea of doing a large study in the treatment of myocardial infarction. And they'd had the support of what was then Stewart Pharmaceuticals, part of ICI, with testing intravenous beta blockade in acute MI. And that was Salim Yusuf's PhD thesis, was looking at the effects of IV beta blocker in the acute phase, the idea being reduce the work of the heart during that period, suggestive evidence of beneficial effects on surrogate markers in terms of infarct size and arrhythmias. And that's when they started talking to Richard about, well, how do we show that that can have a benefit on mortality and morbidity? And as I said, with the collaboration with what became a subsidiary of ICI, the potential to try to do a study at larger scale, kind of create a network of hospitals that could deliver such a trial.
It's funny, isn't it? We just take randomization for granted now, don't we? But perhaps it wasn't so if you go back to that era.
Well, the randomization was certainly very well established. I mean, Austin Bradford Hill introduced it in the 50s with streptomycin. And they're kind of beautiful stories about how he tricked the MRC into allowing the kind of completely unethical idea of randomly assigning treatment because there was only a limited amount of streptomycin. And he writes about how he kind of waited for the right time to introduce the concept. But in a way that was counterproductive because the treatment was so effective for TB that everybody thought we could do lots of small randomized trials and that would give the answer. And the point that Richard has made throughout his career was that you need to randomize to avoid bias, but you need to have large enough numbers of patients, or more importantly, large enough numbers of the health outcome you're trying to influence to be able to reduce not just bias, but random error. And that was really what I came into when I came to Oxford. Was there was just now this opportunity to demonstrate the value of randomizing at scale.
Okay. So I suppose if we go to ISIS-1, which I suppose was that first step, why was it so groundbreaking?
Because it showed you could do a large trial. I mean, in many ways, the treatment hasn't turned out to be particularly relevant. It was a very, very simple trial, so there was no placebo control. And the network was created. So it demonstrated that you could create a network. And again, the thanks to John Cruikshank, who was at ICI then, of introducing us to people in different parts of the world. Sometimes it was the pharmaceutical company representative. Other times it was a clinician they worked with, who then got other of their colleagues in that country to collaborate. And a very good example of that is all the trials done in Italy in acute myocardial infarction. The GISI group grew out of that first network of trialists that formed in Italy to take part in ISIS-1. And indeed, because ISIS-1 went on for so long, Gianni Tognoni and Alan Maggioni and their colleagues decided to stop collaborating in ISIS-1 in order to set up the first GISI trial, which compared intravenous streptokinase versus open control, and was really the kind of the first compelling demonstration that streptokinase could produce benefit in the acute phase of myocardial infarction. And then we went on to replicate that in ISIS-2.
If you look at how trials have developed since then, I mean, I've heard you speak about this many times. Do you think that we've made the whole thing overly complex now? Do you think that's now counting against the development of innovation in clinical trials? Yes.
I think in the mid-1980s, the introduction of the International Committee on Harmonization's Good Clinical Practice Guidelines and the mission creep of those guidelines into the EU first directive and then EU regulation and indeed in regulations elsewhere, has been incredibly damaging. The ICH, you have to look at the history of it. ICH-GCP, ICH is a committee involving pharmaceutical industry and regulators, particularly the regulators in the US, Europe, and Japan, the big markets. And I stress markets because the whole purpose behind ICH-GCP was about marketing. The companies wanted one set of rules that, if they applied those rules, would be internationally accepted for regulatory approval. So it was for the regulation of data generation for new drugs. But it's now been adopted for all kinds of trials. And in 1995, when it was introduced, there was pretty much no CRO industry. There is now a CRO industry which is somewhere between $70 and $90 billion a year. Now, I'm an epidemiologist, so cause and effect is, one has to be a bit careful. But I think there's a very strong association between these rules and regulations with the mission creep and the substantial increase in the cost of the trials. And of course, the incentivization to make them more complicated because you make more profit.
So COVID sort of gave us an opportunity to challenge that paradigm, didn't it? I mean, would you maybe just like to comment on particularly the Oxford experience during COVID?
Well, you say that, but the FDA reviewed all of the trials that were done during the pandemic and came to the conclusion that I think it was more than 95% of them had no reasonable expectation of producing a useful result. So the problem of the regulations, it's resulted in people not being able to do trials at scale. And the regulations really weren't changed during the pandemic. Recovery was the one that got away. It was an exception to really a disastrous set of clinical trials during the pandemic, with, as I say, the vast majority of that activity not being able to provide reliable information. What recovery did was to adopt the ISIS-1 approach. So no placebo control, open label, very streamlined, and yeah, it was a success. It involved all of the hospitals across the UK, but it was very much an exception. And even there, if you look at recovery, and our department ran it, so I'm reasonably familiar with it. The failure to use a factorial design for all of those treatments meant that the results were less compelling and emerged more slowly than they otherwise would have done. So if you look at ISIS-2, the ISIS-2 trial involved streptokinase being randomized, 1 hour streptokinase versus placebo control, and a factorial design, a second randomization of 160 milligrams of aspirin for a month or placebo. And the beauty of that factorial design is you essentially get 2 trials for the price of 1. And the advantage of it is you're always comparing half versus half on streptokinase, half on placebo, half on aspirin, half on placebo. With the platform design of recovery, if you have, say, control versus 4 treatments, you're always comparing 1/5 versus 1/5, not half versus half. And it was only later on in recovery that it was possible to persuade people to factor in convalescent plasma rather than have it as another member of this platform. And that's why that result came so much more rapidly, because it was a factorial design. And the reason why I think there's been a move against factorial designs is, again, going back to the regulations. So the regulations say you shouldn't test more than 1 investigational drug in the same trial. And that has been interpreted as not doing factorial designs. And I think, again, is another area in which it has been damaging and prevented reliable evidence emerging.
So how do we take the renaissance of that design and recovery and expand it? I mean, do you think we heaven forbid, we don't want another pandemic to have to do that? I mean, can we? Is there still momentum behind that idea in developing new trials or not?
I don't know if it's the right word, but to say it's a pandemic, but in the UK, more people died of heart disease than of COVID during the pandemic. So we have a pandemic. we have we're just not allowing ourselves to address the huge numbers of premature deaths and disability from cardiovascular disease, and it is being prevented by regulation. Clyde Meanwell, a Birmingham medical student, graduate who set up the medicines company in the US, did a survey of the US in terms of top 10 drugs. And between, I think, 2005 to 2015, the top 10 drugs resulted in about 2.5-fold increase in income, but about a 7.5-fold decrease in the population that they covered. So what we're seeing in terms of population health is pretty disastrous. The drugs that were coming through are fabulous drugs, but they are drugs for relatively small proportions of the population, and at the expense of doing large-scale population health studies. And the reason for that is you can pick up big effects in rarer conditions in smaller scale trials, less expensive, more rapidly. So from an industry perspective, it makes a lot of sense. But from a public health perspective, it is a really unfortunate trajectory.
Okay. I'm going to pass the baton to Filippo in a second, but just to sort of set the scene there, we've been talking about randomized trials, but a significant amount of your work has been around prospective observational studies. Why do we still need those?
Well, I think what the area that I moved into after the ISIS trials was very much about preventive strategies. So trying to understand major risk factors for cardiovascular disease, and particularly things like blood pressure, blood cholesterol, and body mass index, things like that. The area of cholesterol, I think, really, the epidemiology really helped to inform the design of the clinical trials. Back in the kind of late 80s, I was in China, and we were introduced to a young PhD student called Zhengming Chen, who was conducting a study that was a follow-up to his professor's study done during the Cultural Revolution, where they had measured cholesterol in factory workers. And he was then following up their risks of disease. And the thing that was really interesting about it was that the cholesterol levels of these individuals was way below the cholesterol levels that we were seeing in the UK, the US, and other Western populations. And yet, his preliminary data suggested that lower levels were associated with lower risks of cardiovascular disease. Work that the department had done in collaboration with Cornell across China had also shown between different parts of China that lower cholesterol level was associated with lower cardiovascular disease at levels of cholesterol that we would have thought were not normal, below normal. So it informed the trials in suggesting to us, at least, that if you had a treatment that could lower cholesterol and you wanted to do a trial about the prevention of cardiovascular disease, you should really include people irrespective of their cholesterol level in the West, because they all had abnormal cholesterols. The normal cholesterols were what we were seeing in China, and the lower the cholesterol level, the lower the risk. And at that time, that was pretty revolutionary as a concept, but it did certainly inform the cholesterol lowering trials that we did, the heart protection study, where we really didn't have a lower level of cholesterol. And by testing that, showed that lowering cholesterol, even among people with below average cholesterols for the West, resulted in a reduction in the risk of disease. And really that's led on to the value of adding additional cholesterol lowering agents, even on top of a statin, in order to lower cholesterol further, in order to lower risk further in high-risk individuals. So I think the epidemiology really helps to inform the clinical trials and our strategies for disease prevention.
Great, thanks for that answer, Rory. And I think that's a great point to segue to Filippo. Over to you.
Thank you, Perry. Coming back to observational study, another type of study is about comparison of drugs, treatments, as they say, in the real world, which perhaps is not appropriate because trials are not on the moon, as Milton Packer loves to say. But this study in the real world, do you think they are useful or not? Because definitely it's a different population, much more comprehensive. So if the results of a trial are confirmed in the real world, perhaps this is reassuring. But of course, they are compounders. Putting all together, do you think that there is room for trials, testing, comparing treatments in observational studies?
At the risk of sounding like a broken record, I'm going to go to the perverse effects of ICH-GCP. By making randomized controlled trials that much more difficult, that much more expensive, I think it has driven people to using a poor methodology to evaluate treatment, observational studies, because there is no ICH-GCP that is preventing you from doing observational studies, and so it's easier to do them. We're seeing more and more, rather than reliable large-scale randomized trials, attempts to try to avoid the problems of ICH-GCP and the regulation by using observational data. And the point is that unless you have a treatment that produces a very big effect on a rare outcome, then the problems related to bias, to confounding, are going to make it very difficult to get reliable answers, and more importantly, to know that you've got reliable answers. So for moderate treatment effects on common conditions, I don't think that they are an acceptable substitute to large-scale randomized trials. That's why I go back to Bradford Hill and streptomycin, because really you didn't need a randomized trial. You had a drug that kind of, if you got it, you lived, and if you didn't, you died pretty much. And so, yeah, real world evidence would have been fine there. But if we're talking about treatments that have moderate improvements or in common conditions, then I think you need randomization, and you need large-scale randomization. So, and substituting that because it's too costly and complex to do those large-scale randomized trials seems to me to be the wrong solution to the problem. And that the right solution to the problem is to make them less costly and less complex to do the right thing.
Okay, you've been very clear as usual. Coming back to ICES, I remember that Peter Slide was very proud of this acronym because the ICES is the river of Oxford. But I don't remember who had this idea. Who invented the acronym ICES? Do you remember?
Well, it was just before my time, but I remember being told that it was actually Desmond Julian, who came up with the idea of the International Studies of Infarct Survival and the ICES River. It is, of course, a name that perhaps is not as positive these days as it was.
Peter was very proud of it. Going back to those days, I also remember another quotation about GC from Fausto Rovelli, who was really the mastermind behind GC. And he loved to say, well, to succeed, we must keep it simple. And perhaps he was right, in particular at that time. Simplicity, even in Italy, which traditionally is not so well organized, but even in Italy, keep it simple, really allowed to ran the first trial showing that streptokinase was better than placebo. And perhaps this must be kept in mind from time to time. Not always we have to go complex to achieve good results. Now, as I told you before, I'm speaking from Shanghai. The Oriental Congress of Cardiology is a wonderful congress. Chinese cardiology is progressing quickly. And I'm bound to ask you, what about China Kadoorie Biobank? How did this inform UK Biobank? Can you tell us about?
Yeah, there were a number of people in the UK and elsewhere who felt that we were learning an awful lot about known risk factors by combining relatively small prospective studies. So the blood pressure, observational epidemiology, cholesterol epidemiology. If you pull together lots of observational studies from around the world, prospective cohorts, then one could get really quite clear answers about the relevance of those risk factors for disease. But of course, the problem with that is you could only look at the risk factors that had been known when those studies were set up. And so there were a number of people who were arguing strongly that we should try to create some single, very large scale studies, ideally with blood stored, so that it would be possible to go back to those samples, as well as the questions and the physical measurements in the future, and look to see what it was that was associated with the development of particular diseases. We tried, and I know others tried in the late 1990s to get support to set up such studies, but there really didn't seem to be much enthusiasm. And we talked with lots of people to see whether there were opportunities to set up such a study. And through a colleague in Hong Kong, who we knew through her work on tobacco activism, we gained an introduction to the Kadoorie family. The electric company and various other property companies in Hong Kong. And they agreed to provide funding to set up the China Kadoorie Biobank, a collaboration that we were putting together with the Chinese Academy of Medical Sciences in Beijing. My colleague Zhengming Chen, who was that PhD student that I mentioned earlier, who had now moved to Oxford, worked with the Chinese Academy of Medical Sciences and the Centers for Disease Control in China to select 5 rural and 5 urban areas across China that fulfilled a number of criteria. One, that the exposures, the lifestyle of the people in those different populations would be very different. And 2, that those Centers for Disease Control were well established for following disease, and particularly death and cancer, which was all that was expected. So that's how they went about setting up the recruitment in those 10 regions of 500,000 people aged between about 40 and 80 years of age. We were then incredibly lucky because the Chinese government introduced a health insurance scheme that pretty much everybody in China signed up to, which is a single electronic healthcare system that allows us to get much more follow-up data for the Kadoorie Biobank than we had anticipated, because the people in China use that system, and you can get a lot more information beyond the death and cancer data that we had anticipated, and that's proved to be fantastically valuable. So it was set up just before UK Biobank, but I happily stole many of the ideas of how Zhengming thought through the way of recruiting and assessing participants in UK Biobank. So what he was doing was setting up assessment centers in different parts of China, and then people would be invited to attend, and then they would move through different stations in that visit, from questionnaire to physical measurement to biological sample. And we really copied that in the way that we managed the UK Biobank project when I was asked to take it on in 2005.
Fascinating story, really, really very informative and inspirational. What has been the most rewarding aspect of the biobank so far?
Well, when I was asked to take it on, the Wellcome Trust and the Medical Research Council, who funded it and founded it, really, there were very few conditions, if you like, of what they wanted. They'd had a group set up to think out an outline protocol that was led by Tom Mead, and they wanted to recruit about 500,000 people, men and women who were in middle age. And the key thing they wanted was that the data would be readily available to researchers everywhere in the world. And so I think the thing that I've found most exciting about UK Biobank has been working out how to make the data accessible to researchers around the world. The Kadoorie Biobank has, because of various constraints on data access, not had that benefit. We're still working towards that. But with UK Biobank, making the data accessible and allowing the imagination of scientists around the world to use it has been absolutely fascinating from my perspective. If it had been one research group using the data, they would have been using it in one particular way. But by having lots and lots of imaginations, approaching the data in completely unexpected ways, I think so much more has emerged from it. And as a consequence of that, organizations have been willing to put more resource into it to make the data more valuable. So we've seen the Medical Research Council and Wellcome Trust funding enhancements of the resource, such as imaging, that are making the data even more valuable for studying various diseases, be they diseases of the brain, the body, the heart. We've seen industry come in and start to fund assays of the samples because that makes the resource of more value to them, but then those data become available to the global research community. So just observing how sharing data has been beneficial to everybody, including the team building it, has been a kind of fascinating experience. And it has also occurred at the right time, whereby we're seeing publications or pre-publications going onto the internet that allows researchers to say what they found, but allows others to look at what they found and check it. Almost pure form of peer review. You know, when you get a paper that's sent to a journal and reviewers are having to look at what's written in the paper, they can't really check what's happened. But what we've seen is some really nice examples of articles with fascinating findings going onto medRxiv. Other researchers who are using the UK Biobank data saying, that's an incredible result. I should see whether I can replicate it. And sometimes finding errors in the analysis that have then been corrected by the original authors prior to publication. So I think that's been a kind of really extraordinarily positive collaborative enterprise between different research groups.
And what is more challenging in the biobank?
The biggest challenge, I think, is to maintain that, to maintain accessibility. I think the biggest personal challenge I'm facing is how do we get access to all of the health outcome data? So at the moment in UK Biobank, we get linkage to death and cancer and hospitalization data because they are available centrally within the NHS. The primary care data in the UK is not. It's actually the data controller are the primary care doctors. So it's not available centrally. And so we're working very hard with the Royal College of General Practitioners, with the British Medical Association, with NHS itself, to see how we can honour the consent of the participants for the primary care data also to be available. and to extend the linkages to other kinds of data, to registries of imaging data, of joint replacement data, in order to enrich not just the characterization of the participants, which is what we are doing with, say, imaging or sequencing or proteomics, but increasing the depth of characterization of the diseases that the participants are now having, in order that we deal with the major weakness of prospective studies, and that is that the participants are often characterized in great depth, but their disease is not. What we want to do is combine depth of characterization of exposures with depth of characterization of specific diseases, and that's really where we're trying to shift our emphasis.
Thank you, Rory. Now I have a question with Perry as editors of the European Journal, because every week we receive at least 5 papers based on UK Biobank data. And if we accept a paper, which makes a lot of sense, well presented, innovative data, of course, the risk is that other authors publish the same paper based on the same data in another journal. And this can really create confusion also because not always, not necessarily the results are similar. How can we defend ourselves from these duplications?
Well, I mean, my feeling is that this meta-archive approach is helping in that respect, because one can see what is coming down the pike. So the opportunities to, first of all, look at what is being done in UK Biobank, what is emerging and might go onto meta-archive, and then look to see whether one can scope that out and help to inform journals. Whether that's done by us, whether that's done by the journals themselves, or some other kind of organization to try to coordinate this. But it's a nice problem to have, in the sense of having lots of really interesting findings emerging rather than the alternative.
Yeah, duplications better than nothing. This is your message. And anyway, we have to keep an eye on meta-archive. This is your suggestion. Now, before asking Perry, I'm sure he has some other questions. Our audience is very excited about this dialogue. They find it, as expected, very inspirational. I have 2 questions. One is from Marius Roman. The question is about the increasing number of adaptive design trials. What is your attitude? The second is from another colleague, Dr. Seb Kar, and is about pre-specified or non-pre-specified subset analysis. We know you're very critical about it, but can you tell us about?
Well, again, I think if one looks at the so-called platform trial of recovery and compare that with a factorial design, or even a partial factorial, if there are treatments you can't give in combination. The problem with adaptive designs with a platform study is that, let's say you have 4 treatment groups compared with a control, and the results become compelling for one of those treatment groups, and therefore you stop it, as with steroids in recovery. Then your ratios of treatment to control change. And therefore your analyses become have to be stratified, because you have different ratios each time you drop out a treatment or each time you add in a new treatment. Whereas with the factorial design, it's always half versus half. And there's a very nice example of that. The Ascot trial, which was a factorial trial in people with hypertension run by Neil Poulter and Peter Sever, a factorial trial of statin versus placebo and of different blood pressure regimens. The statin result emerged really rapidly, and the data monitoring committee said we've already got a clear result, and they stopped the statin arm, and those results could be used by clinicians, but it had no impact on the blood pressure comparison, which could just continue on. So I think adaptive designs in that respect, in a factorial design, work very well. in the so-called platform trials, which, because of recovery, I think have become quite trendy. I think actually it's not a great design. And as I say, you can do partial factorial studies whereby if there are some treatments you can't give together. People argue against factorial designs because they say, well, what if there are interactions? To which my answer is, if there are interactions, a factorial study is the best way to find them out, rather than find them out down the road in your real world. So again, I think if treatments are going to be given together, then giving them together in a factorial design is a good way forward. With respect to the second question of pre-specified analyses, again, gosh, really is broken record time. But if you go back to ICHGCP, there is all this emphasis about how the subgroup analyses have to be pre-specified. So what do people do? They pre-specified very large numbers of hypotheses, which just completely undermines the whole point. If you think there is a good reason for there being a differential effect, then fine, pre-specify it, but also specify in what direction. I've rarely seen examples where, with all these subgroup analyses by age, gender, this, that, and the other, it actually says, well, we are anticipating a particular direction of interaction. In ICES 2, we did pre-specify that we thought the earlier you treated, the bigger the effect. But we pre-specified not just what the subgroups would be, but what the direction of effect was we were hypothesizing. And again, the idea that you just pre-specify dozens and dozens of subgroups completely undermines the whole point of it.
Well, very good reasons to reject papers if this the result, the expected result of the pre-specified analysis is not provided. Very good point. Thank you, Rory. Perry, to you.
Yeah, thanks, Rory. You've just made the editorial committee meetings just that little bit more difficult. Thank you.
Always glad to help.
So, I mean, there were a number of specific questions we sort of lined up for you, but I was going to ask you a more general question. It's being said that academic medicine's in crisis, that we've got fewer people coming into it. Those that do come often don't stay. I'm sure you read that BMJ leader a few weeks ago calling for action on this. Do you agree with the fact that there is a problem? And if you do, what can we do about it?
Well, I think there's a problem, and there's also kind of an opportunity, if you like, but it may not be the one that academia particularly likes. I mean, go back to my original point about the difference between mentorship and sponsorship. It takes an awful long time before you can get going in research. The level of support people need isn't there. I think it's much tougher now for a young person to get into research and continue in research to the point where they can demonstrate that they can do it. I was phenomenally lucky, and I think you'd have to be even luckier now to get that opportunity. So I really feel that we need to be investing in career track, supporting people during those early years to a much greater extent than we do. And that we need to look at how we do that, both for men and for women. You know, we see real drop-off in female academics, and we've made an enormous investment in them. And then at the point where they're starting to become kind of return on that investment, we don't provide the extra support that's needed. So I feel that there needs to be a real redirection of resources to support researchers at the early stages, and to look at what are the obstacles to that. When I look at the opportunities now, I kind of think, well, would I go into academic medicine, or would I have a look at some of these really interesting companies that are emerging, which are kind of quasi-academic. I mean, I've worked quite a lot with Regeneron, with UK Biobank, who've done very well in recruiting from academia. And those academics in Regeneron or companies like Regeneron are being given opportunities to do really good research that they would struggle to get within the academic setting. And so you have to ask the question, if you want to do research that makes a difference, is that in universities as they are now? Or is it in some of these discovery science companies, like the Regenerons of this world or Calico? I'm sure there are many others that people are familiar with, where you get to do really good science, you have the resources to do really good science, and you can collaborate with academia. And again, I think increasingly fantastic collaborations with them. So increased porosity between academia and those kinds of companies, I think, is very attractive. And then what we need to do within universities is work out how we stop that porosity, resulting in the traffic being all one way and people moving to the companies. And what are the things that we can do? And I think it goes back to, we need to support our younger researchers far better than we do.
Mm. I mean, if you look at our own situation in the UK, we have a lot of money. As much as we moan about it, we have a lot of money in the system, which is theoretically there to support research. So the resources are probably there. Who is it that's going to drive this change? Is it going to be research funders? Is it going to be the universities? Do we have to completely come up with a new career path, career structure? Is the reward, the incentive system the right one to encourage people to come in and stay? I mean, we talk about it a lot, but how are we going to change this?
I'm
sharing the pain. I'm sharing the pain, Rory.
I think that it's going to have to come from the funders, because it is about money. I think people in research need to be saying to the funders that if we don't have a pipeline, then the whole thing will fall over. So although I'm in a situation of getting very substantial funding to run UK Biobank, what I think it's also doing, by going back to my point about accessibility, is it's making opportunity available to smart young researchers to do research that is as good as anybody else. They don't have to create the cohort. They just have to be smarter than the others and to know how to use the systems better. And so I think that there is the opportunity there, and this is a kind of move towards open science, the opportunity to use data that exists rather than having to build your own data in those early years, does provide a real opportunity for younger researchers to do stuff better than older researchers, because they can focus on it. And therefore, I think the funders should take advantage of that by making sure that they don't spend all their time scrabbling around trying to get small grants, but actually look after them for a period of time and give them an opportunity to demonstrate that, that they can do it.
It's almost like a research apprenticeship in a way.
Yeah, and when you think about a lot of PhDs that are done, that the value is about training, not necessarily about what comes out of the PhD. What I've seen with UK Biobank is that there are people who are now doing PhDs where there's enormous value that comes out of it. I mean, for example, the whole concept of polygenic risk scores was a PhD student at the Broad, Amit Kira. His PhD was on was essentially inventing the whole concept of polygenic risk scores and demonstrating that they could be used to predict people at high risk. And I'm not saying that UK Biobank is a solution to all problems, but I think the idea that people don't have to generate their own data, but that they can access data and do really unique things with it, provides a much faster opportunity for them to demonstrate their ability.
Thanks. That's a really interesting idea. Filippo, I'm conscious that we're sort of approaching the top of an hour now, so maybe I could just hand the baton back to you.
Well, I have some interesting questions from the audience. One is from a colleague from Oxford, Dr. Nazar Zadeh. The question is what about the opportunity to design a nested trial within the UK Biobank? Is a possible perspective?
Yeah, so UK Biobank doesn't have feedback, but in fact, a hypertension trial did recruit through UK Biobank, the TIME trial. So this was a question about when to give blood pressure lowering treatments. And so we did write to people in UK Biobank and said if they were interested in being involved in the trial, they should contact the investigators. The issue that UK Biobank has, as I say, is there isn't feedback, and this is really what Our Future Health is trying to address. The idea that you could recruit people on the basis of particular characteristics, be that genetic or certain other factors that make them particularly eligible for a trial. But I mean, actually, in the UK, and of course, beautifully demonstrated in Sweden with Sweetheart, one can use national or regional health record systems to identify people with particular conditions and then recruit them directly into randomized trials. And as I say, Lars Valentin and Stefan James have demonstrated this beautifully within the Swedish system, and more recently, very nice studies of different screening strategies in the Danish healthcare system. So I think our healthcare systems, particularly as they get more joined up, and if they become accessible for recruitment, they can be used, I think, to facilitate recruitment into trials. But of course, if the patients are embedded within a healthcare system, they can also be used to follow up those patients, not just during a particular trial period, but in the long term to find out whether there are unexpected both benefits or harms. And again, beautifully demonstrated by the WASCOP study with statins, where they showed the beneficial effect of statins on heart failure only many years after the active part of the trial ended.
Thank you. One last question from Franz Messerli about meta-analysis. At EHJ, we prefer patient-level meta-analysis rather than study-level meta-analysis, and we're always a bit worried about network meta-analysis. What is your attitude?
Well, my main comment about patient-level meta-analyses is they're a heck of a lot of work. I mean, getting the individual patient, they are a lot of work to get the individual patient data. On the other hand, if you do get it, then it allows you to address a lot more questions in a lot more detail. But you know, streptokinase was tested in acute myocardial infarction because of a tabular meta-analysis that really indicated that there was likely to be a reduction in mortality of about a quarter. And that's what GC confirmed, ISIS-2 confirmed, and then many subsequent trials confirmed. So they have value, and they can give you an idea of really promising areas to then confirm.
Do you trust network meta-analysis?
Well, I think that the idea that you can kind of compare A versus B and then B versus C and work out what A versus C, yeah, I think that these are provided you have sufficient data, provided you have the depth of information about those studies. I think again it has merit, but one needs to be careful in how definitely you interpret it. And I think a lot of meta-analyses probably need confirmation. So I would use them a lot of the time for indicating directions of travel for future studies.
Thank you, Rory. I have one final personal question. Your wife is Barbara Casadei, past president of the European Society of Cardiology. By the way, during COVID was a dreadful time and she did extremely well. She's a top scientist. She has been with us for another dialogue before. And so you are the most famous couple in the field of cardiology. My question is, what is your favorite topic at home?
Well, I guess at home, if you like, I'm interested in house and she's interested in garden. Barbara's very interested in living things, particularly in the garden, animals, people. I guess I'm more concrete. I love the idea of design. So I guess that's our kind of complementary approach to home, but I think also a complementary approach to science. I mean, she's very much kind of basic science translating, whereas I'm going from translation into population. And I think those kind of complementary strategies have been very positive. And I think that's how research works in general very well, where people with complementary strengths bring something different and work together effectively.
Well, let's close with this very important remark. I remind our audience that next dialogue will be as usual on the last Thursday of the month, will be July 25th at 25th, and will be at 17 Central European Time as usual. Thank you, Rory. Thank you, Perry. It's been really a very interesting hour, interesting questions. And Rory, I hope that you enjoyed our dialogue. Do you have suggestions for our dialogues? What can we improve?
No, I think it was, I very much enjoyed it. I hope others enjoyed it as much as I did. But thank you very much for the opportunity to have a chat.
Thank you, Rory. Yeah, the audience, the feedback from the audience is extremely positive. Thank you.
Nice, thank you.
Thank you very much, Rory. Thank you, Perry. We'll stay in touch.